Okay, good afternoon. My name is John Garen. I am the Chief Business Officer with uniQure. We are a publicly traded company on the NASDAQ with the symbol QURE, and this is a standard forward-looking statement, disclaimer, and this is applicable to this presentation. So uniQure is an AAV gene therapy company pursuing the promise of single treatment of severe diseases. We've been around for a long time. We were once Amsterdam Molecular Therapeutics, or AMT, and became uniQure and became publicly traded a few years back. We have assets really in four areas. We have programs, we have technology, we have intellectual property patents and know-how, and we have a GMP large-scale manufacturing facility in Lexington, Massachusetts that's online. So I will cover right now the programs, and our lead programs really fall into these four areas. The first is our hemophilia B, which is advancing into a pivotal trial and has completed a phase 1/2 study. Our Huntington's disease program is completing preclinical development, and we intend to bring that to the clinic in 2018. And we have a cardiovascular diseases collaboration with Bristol Myers Squibb, with the lead program there being S100A1 for the treatment of congestive heart failure that we are advancing through preclinical development. What I think is very interesting about these three areas is that they represent three distinct applications of AAV gene therapy. One of them is a knock-in approach with the hemophilia B, where you have a mutation leading to a defective or non-functioning protein, and you restore the function of that protein by delivering the DNA for a normal functioning protein. The second is Huntington's disease, which is a knockdown. You have a mutation leading to a deleterious protein, which then causes the disease, and we then treat the disease by delivering the DNA that produces a microRNA. The microRNA interferes with the messenger RNA of the Huntington protein and thereby ameliorates the disease. In the last instance, with S100A1, you don't have a mutation-driven disease in congestive heart failure, but you have a therapeutic protein that has benefit in the disease, and we deliver the gene therapy for that protein to the heart and achieve some benefits from that purpose. So it's a therapeutic protein delivery system. So just looking at the progress across the company in 2017, I would definitely draw the attention to the very first point where we achieved breakthrough designation with the FDA and prime designation with the EMA for hemophilia B. And these were certainly an important achievement for our regulatory organization, but also a strong endorsement of the fact that this disease is a serious unmet need and that we have a valuable treatment for it. And I will go through the various different progress we've made in the different collaborations and other indications. In the last instance, we have generated data with our proprietary AAV5 vector, which really distinguishes it from all the other vectors in development, we believe, in terms of its immunological profile and its ability to provide sustained benefit, which has not been seen with others and is very distinctive for this one. Some of the achievements to date that we've had just in recent announcements, we've achieved scale-up manufacturing at GMP quality for our Lexington facility. We have advanced the hemophilia B program in both CMC and regulatory areas, and we have gotten a patent in our manufacturing IP portfolio to issue, which added to many other patents, establishes us as really a solid leader with a strong proprietary position in our manufacturing platform, which is insect cell baculoviral-based manufacturing of AAV vectors. And I'll discuss that more in detail. Now, to just touch on again the AAV5 vector, this is a proprietary vector for uniQure, and it has been shown in numerous clinical trials to be effective and able to deliver the benefits necessary. It also has demonstrated the lowest level of pre-existing antibodies against the vector, which is very important because you may understand that when you have pre-existing antibodies, those patients will get excluded at a certain level of those antibodies as candidate patients, which takes them out of the target population. In our case, that number seems to be very, very low, which makes us able to treat a larger number of the target population. We've also demonstrated very good delivery and the fact that we don't see a T-cell response against this AAV vector, and so it gets long sustained benefit. And you may know that some programs have been terminated simply because they were not able to get sustained benefit. So I will show you how we've been able to overcome that. Now, turning to our manufacturing facility in Lexington, Massachusetts, it's a 55,000 square foot facility that's state-of-the-art GMP manufacturing, end-to-end of AAVs, and we're able to deliver upstream and downstream process, drug substance, and final drug product, and everything else associated with it. So it's a very beneficial and compelling asset for us. The features of this system, as I mentioned before, we work in insect cell baculoviral-based manufacturing. We are on a third generation and continuing to innovate that platform. It is very well IP protected, and it has up to 4,000 liter scale manufacturing capability. Now, some things that have arisen in discussions in the past are around the benefits, or how do you compare insect cell-based manufacturing to mammalian cell-based manufacturing? And we've chosen to take the insect cell-based approach for several reasons. We believe it is a superior platform for commercial scale manufacturing for really several reasons. Here are some of the key ones, which is the fact that it's highly scalable. And it's because you don't have some of the complex transfection processes that you see with mammalian cells. There's not a centrifugation step that's required, which is a very difficult step to scale. It is also quite safe because you use insect cell promoters and not mammalian promoters. You don't have the risk of replication-competent viruses getting into your product, and we don't need to use mammalian serum or any serum at all, and therefore you avoid some of the contamination risks associated with that. And then lastly is the cost effectiveness of this platform for the reasons above, as well as the fact that a lot of the materials that are used in this platform are not nearly as expensive. So this is an excellent commercial platform for manufacturing of AAV. Now this is a snapshot of our pipeline, and I will take us through the hemophilia, the Huntingtons, and our cardiovascular projects. So beginning with hemophilia B, first, to reiterate, we've achieved the both FDA breakthrough and prime designations from EMEA. All right. So we've completed a phase 1/2 study with this, 10 patients, 2 cohorts, and 9 European sites, and this was conducted in adults with severe or moderate to severe hemophilia B that are taking prophylactic factor IX and have bleeding events. This is just an overview of the baseline characteristics of these patients, and I would just note that the low-dose cohort was a bit older and more symptomatic than the higher-dose cohort. And what we've seen is very impressive clinical benefits from this trial in several areas. The first area is the reduction in the need for factor IX, recombinant factor IX therapy by 85%, which is certainly very compelling. We've also seen an 84% reduction in bleeding events, which is another a significant clinical benefit on efficacy. And then we've also seen now sustained factor IX activity levels over the course of 1 year for all the patients. And again, that is a very critical attribute of our AAV5 vector, which enables patients to benefit from this without dropping out. The product was generally safe and well tolerated, with some adverse events listed here which are manageable. And again, the immunological profile, we saw no evidence of any T-cell activation, and we did not see any generation of factor IX inhibitors, which is a very important quality of what we've done. So, as I mentioned before, several programs were terminated because they did not have this ability, and what we've seen is an excellent profile with this particular vector. So the next steps of the hemophilia B program are really to bring it into a pivotal trial, and we are working diligently to do that right now. The next program I will discuss is our Huntington's disease program. So as some of you may know, Huntington's disease is an orphan indication, but it's fairly prevalent with a very high level of unmet need. We have demonstrated very compelling preclinical data in large animals, and I will show you some of that. And we are working right now diligently to bring this to first-in-man studies in 2018. So this slide summarizes some of the preclinical results that we've seen with the knockdown of the messenger RNA for the Huntington protein as well as the mutant Huntington protein levels themselves in a large animal mini pig model. And you can see, particularly when it comes to the mutant Huntington, that it is widely distributed in the brain, which is important, and it's also nicely dose-dependent. So we see this as a very compelling and promising basis for bringing this into the clinic. The next area is our cardiovascular diseases area. And here we have a quite productive collaboration with BMS. That was a landmark deal for uniQure. We have, as a lead program there, S100A1, with the potential to bring 10 targets total forward under BMS. The economics of the deal were very impressive and very beneficial for us, with $2.3 billion in potential milestones and significant royalties. BMS has taken a significant 10%, nearly 10% equity stake in the company, with warrants to go to almost 20%. Progress with this program has been quite good. I won't necessarily go through with you the congestive heart failure space, but it's a very significant area. We have generated very good data in terms of delivery of gene and protein expression within the myocardium. We've also seen very good preclinical results for the program, and right now are moving along in our preclinical development with an effort to bring this into the clinic as soon as we reasonably can. So just lastly, in terms of our goals for 2017, much of it I've already covered. We're taking forward our hemophilia B and advancing into the pivotal studies, advancing Huntington's into IND filing, advancing our heart failure program into the IND enabling studies that we'd like to initiate for our IND filing. We are presenting new data as we've described on the programs. We've completed our manufacturing and scale-up campaigns in Lexington, and we intend to try to announce new areas of indications that we will be pursuing this year. So that concludes the presentation, and I'm happy to take any questions that you may have. Thank you.